Okay, good afternoon, everyone. It is my great pleasure today to introduce you to Professor Marco Bianchi, who's an eminent Italian scientist whom many of us know since a very long time. Marco, I guess, graduated in Milan, then he spent quite some time abroad at Yale University, and then also quite some years in Heidelberg at the MBL. After this long stint, he returned back to Italy, first in Pavia and then at the University of Milan, when I had the pleasure to first get to know Marco. At the time I was a student there. And subsequently he moved to the University Vita e Salute San Raffaele and to the DBIT. And since then he's working there, where he's also professor in molecular biology at the Faculty of Medicine. Many of you know, and I guess you can also imagine from the title of his talk, that Marco is renowned worldwide for his pivotal contributions in the field of high mobility group box proteins. And without further ado, I leave the floor to Marco. And thanks for being here, and we are looking forward to your seminar. Thank you.
Thank you for listening to me. This seminar covers a multi-decade story, so it will be a fairly complicated story. And just to let you orient, I have also introduced a timeline where the story actually starts before I run into HMGB1. So HMGB1 stands for High Mobility Group Box Protein 1. And actually it was identified already 50 years ago as an abundant protein in the nucleus. And that was it. And in 1989, our group, which at the time essentially was me and another person, defined that HMGB1 bends DNA. And so this is the main function of HMGB1. It works as a chaperone for DNA. It bends DNA and then leaves. Okay? Then someone else in 1999, our friend and competitor Kevin Tracy, showed that HMG1 is secreted by macrophages during sepsis. That at the time looked crazy because HMGB1 was a nuclear protein and there was no way a nuclear protein could be secreted. And then we realized that just because of this, that HMGB1 is a damage-associated molecular pattern. What is a DAMP, I will define later, and the trigger of inflammation. And then, more or less at the same time, we realized that HMG1 is an adjuvant for immune responses. And then we've realized that the whole story was too complicated and didn't make sense, because in fact HMGB1 is at least two different proteins that have different effects, because it has two different redox forms. Actually it has three, but the third is inactive. And so the reduced protein works in a way, and the disulfide containing protein works in a different way with different receptors. And so much of the confusion was due to the fact that as if you have two pictures of two different people and you superimpose them and you don't understand anything. And then we realized we actually hypothesized and then demonstrated a few years ago that HMG1 orchestrates tissue regeneration. And more recently, we have are showing that HMG1 is a pivotal protein in a form of cancer immunosurveillance, and so potentially can be used for cancer immunotherapy. Okay, let's start from the beginning. So the background is the danger theory. So essentially, it was shown already in the 70s that our organism recognizes pathogens thanks to pathogen-associated molecular patterns, which are molecules, which are broad classes, essentially, that are recognized by specific receptors, mainly on macrophages, but mostly in dendritic cells, that recognize these groups of proteins via the receptors, which are toll-like receptors mainly. So a dendritic cell sees one of these molecules and the toll receptor is activated and the dendritic cell activates short-term and long-term immune responses. But the same thing happens also with damages which are not caused by infection, but simply injury. So there are forms of injury where there is no infection, and this is called sterile inflammation. And these are, sterile inflammation is due to damaged cells that in some way, due to mechanical damage or the damage produced by some pathogen, are broken up and release what are called damage-associated molecular patterns, or DAMPs, which are proteins of the cell, which are usually inside the cell, and so cannot be seen by external cells and specifically by cells of the immune system. But when the cell dies, this interior of the cells spills out, and so the immune cells see the internal proteins and understand that something is amiss. The usual comparison I make is that if you see blood on the curb, you know that someone has got injured. You don't know what happened, but at least you know that something is wrong, okay? And this is what happens. So these cells release their proteins, and these proteins, which are usually inside the cell, now get visible to the dendritic cell and the macrophages that now activate, again, short-term inflammatory response, but also long-term adaptive immunity responses. Quite interestingly, the receptors are mostly the same. So some toll receptors, you will see, recognize both PAMPS and DAMPs. And the outcome is very similar. The most significant difference is that the adaptive immunity processes activated by DAMPs are usually short-term. So if they are not sustained, they just go down. While adaptive immunity, which is caused by PAMPS, usually are long-term and produce antibodies and T-cell clones that persist. So at that time, essentially, Pauline Matzinger had just proposed her danger theory. And so the danger theory states that the immune system does not really recognize self from non-self, because in fact, it does not have a real list of what is self and what is non-self. So without having a list, it cannot do it. But essentially, it recognizes antigens, all antigens, but distinguishes between the ones which are exposed in a danger situation, so are potentially antigens that are associated to some causes of danger to the organism, and antigens which are exposed in a situation of calm, which then means they're not relevant. Okay? So, especially for antigen-presenting cells, this means that the antigen-presenting cells, of course, like the name implies, show antigens. They expose them on MHC class II molecules. The T cells recognize them, but then do not respond. Or if they respond in a tolerogenic way. But then, if the same cells, in addition to antigens, see adjuvants, so other molecules that activate this sense of danger, then express other molecules that help create a synapse between the antigen-presenting cells and the T cells, and now produce a long-standing response in T cells, which is stimulating. And so the T cells now produce an immune response that ends with the production of antibodies and T cell clones, which are effector. Okay, so so far so good, but this was only a prediction. And so the cause of the danger signal, so the molecular actor of the danger signal was not known. So there was a first sighting that turned out not to be correct. So it was first identified as uric acid. And then essentially at the same, almost at the same time, we came up with HMGB1. So it was a fairly chance finding. And it's only because we were working on HMGB1 that we realized that HMGB1 is the the danger signal that activates the adaptive immune response. So what is HMGB1? HMGB1 is a protein which is very conserved across evolution. It's present in all eukaryotic cells, including yeasts and including unicellular organs, paramecium, and so on. It's not present in bacteria, and it's really very conserved. So you can recognize it even in unicellular organisms, and it's identical in all mammals. So there are only conservative substitutions between aspartic acid and glutamic acid and vice versa, which is convenient because you can use the human and the mouse protein just in the same way because they're identical, essentially. The main function of HMG1, and that was our interest at the time, is that HMG1 is an abundant chromatin component, and we showed that it bends DNA, and in fact it chaperones DNA bending, and it's also involved in the formation of nucleosomes because nucleosomes are deposited on DNA by chaperones for histones, but there is also a chaperone for DNA, which is HMGB1. So this is the essential function of HMGB1. It's a simple protein. It's only 215 amino acids. It contains two structural domains, which are called HMG boxes, and are called the box A and box B, which are linked by a flexible linker. And then there is an extended unstructural domain, intrinsically disordered domain, which is mostly acidic. It's 30 amino acids, which are all negative, creating a very long tail. So this is a fairly reasonable representation. This is the size of the domains, and this is the size of a tail. So the protein is completely non-globular, okay? Each of the domains can bind the DNA in the minor groove, and that's how it bends DNA, because it enlarges the minor groove and the DNA bends. So inside the nucleus, the function is to allow proteins which are bound to DNA come close when the DNA is bent and form macromolecular complexes which are producive to what you need. Okay, so generally it's involved in transcription factor binding, but also in recombination and so on. But the story became interesting when Kevin Tracy found that the protein is released by macrophages during infection and sepsis. So we were bemused because we knew that HMG1 was a nuclear protein. And so a nuclear protein cannot be secreted simply because it does not have a signal peptide that delivers it to the endoplasmic reticulum, so it cannot go to the secretory vesicles. So we thought that there must be something related to this. We redid the experiments, and in fact it was true. HMG1 is released during infection by macrophages. So we thought that maybe the protein was released because it had a function when it is released, which is related to something that happens spontaneously. So we knew that HMGB1 was binding to DNA, but very reversibly. So we thought that maybe the protein was involved in cell death and would be released upon cell death. So we actually tested the two forms of cell death, which the two forms that were known at the time. We are talking about the early 2000s. And at that time, the cell death was divided in necrosis and apoptosis. So necrosis was everything, and apoptosis was the specific cell death where the cell kills itself by a very stereotyped program which depends on caspases. So we actually showed that if the cell goes into apoptosis, HMGB1 binds to the chromatin and is not released. Whereas if a cell dies by necrosis, which in this context you should take as non-apoptosis, the cell releases HMGB1. And so the HMGB1 can be seen by other cells because now it's outside of the cell. So this was quite simple. Now the story is a little bit more complicated, but the sense is the same. The HMG1 is released in very many types of cell death, all cell deaths which are not apoptosis. These are only some of them. Excitotoxicity is overstimulation of neurons that then say goodbye and die. It's pyroptosis, which is the overstimulation of macrophages that then explode and irrorate their surroundings with cell components, including HMGB1. HMG1 is released over infection by viruses that have anti-apoptotic proteins. So the cell cannot go into apoptosis, but it dies anyway and releases HMGB1. But it's also due to death because of alkylating agents or because of excessive reactive oxygen species, and so on and so forth. Every year or so, a new cell death mechanism comes out. So HMG1 is released upon a ferroptosis, is released upon cuproptosis, everything except apoptosis. But the initial finding was enough to tell us that maybe this was the signal that was signaling to the immune system that something was amiss. Because if the cells were killing themselves by apoptosis, they did not release HMGB1, while if they were dying because of other causes, they were releasing HMG1. So there was a cue for the immune system to distinguish between a cell dying in a neutral way and a cell being killed by something that was not right. So we actually showed that HMGB1 activates the immune system. So essentially, this was a critical experiment. If you inject lymphoma cells into a syngeneic mouse, the lymphoma grows, and there is essentially no immune response against the lymphoma cells. But now, if you inject the lymphoma cells together with the supernatant of cells which have released HMGB1, so it's simple, you just take the cells and freeze and thaw them and take the supernatant, and you inject it with the lymphoma cells. Now the lymphoma cells are rejected, and you have tumor-free animals. This only if you inject the lymphoma cells with wild-type cells. If you do the same with cells which are HMGB1 knockout, then the lymphoma essentially grows and the tumor-free animals are fewer. If you just inject lymphoma alone, all the animals die. If you inject PBS, the number of inject the lymphoma with PBS, the number of the animals which are tumor-free is essentially zero. So you have a response against cells which are very lowly immunogenic if you add HMG1, which is exactly what was the prediction of the danger theory. The same thing happens with soluble antigens. If you inject ovalbumin alone, you have a certain response. If you inject ovalbumin plus HMGB1, now the response is much bigger, and all mice respond even to a very low amount of albumin and produce antibodies. So this is exactly what was predicted. Now, the story now became more complicated because, of course, it turns out that the experiment we did reproduce a situation where HMG1 is released essentially by dead cells. But the initial story that was reported in Science by Kevin Tracy showed that macrophages secrete HMGB1. So how can you secrete HMGB1? So it turns out that there is a passive release and an active release. The passive release is that simply you just break up the plasma membrane, the cell membrane, and the HMGB1 flows out. The active secretion is much more complicated, and at this point I believe, but I do not have the proofs, that there are several ways to secrete HMGB1. One has been clarified rather well. In this case, you have a virus, for example, which activates a cell. The cell now has NF-kappa B activated and produces interferon. The interferon goes out and binds to the interferon receptor. The interferon receptor now phosphorylates STAT proteins. The STAT proteins go to the nucleus and start to work as transcription factors, activating specific genes. But one of the things that STAT proteins do is they also recruit acetylases. And so the acetylases which are recruited now start working on all nearby proteins. So they acetylate histones, but they also acetylate HMGB1. By acetylating HMGB1 on the nuclear localization signal, HMGB1 can now go out of the nucleus, but cannot go back to the nucleus because the nuclear localization signal is obliterated, is not recognized any longer. And we showed that HMG1 can go to the lysosome, and then the entire content of the lysosome can be secreted or released outside. So this is an active secretion. The cells that release HMGB1 actively are not going to die. So they live without HMGB1 for quite some time, and essentially they take their time to reproduce HMGB1. So the whole process is completely non-transcriptional in the sense that the HMGB1 which is secreted was already there. It goes down to almost zero, and it's only reconstituted 2 or 3 days after the fact. So the cells do not die without HMGB1, they make it later. But it's not only viruses that cause the release of HMGB1. The cells under severe stress, for example, hypoxia, secrete HMGB1 because they do the same. They first acetylate the nuclear localization signals, HMG1 goes to the cytoplasm, and somehow it goes outside in the medium. And this happens also in reality, not just in cell cultures, because people showed that during ischemia reperfusion of a heart, HMGB1 is secreted by cardiomyocytes. Then the protein recruits inflammatory cells in the heart. And in fact, if you use an inhibitor of HMGB1, so this is box A, there's a fragment of box A which competes with real box A, sort of fake box A, HMGB1. So in this case, there are far fewer cells which are recruited. Of course, this does not happen only in ischemia reperfusion of the heart, but other people showed that it happens in brain ischemia, in hepatitis, in peritonitis. Whenever you have some tissue damage, the cells are activated to secrete actively HMGB1, and HMG1 is the signal that causes the recruitment of inflammatory cells. It's not the only one, but it's the main one, at least the one which is more general. So the question is, how do cells migrate towards HMGB1? We were looking for a receptor, and we couldn't find a receptor. And then we realized that maybe the receptor would be something which is very commonly expressed. And the most commonly expressed receptor in the body is CXCR4. CXCR4 is a G protein receptor. Already G protein receptors are almost everywhere, but CXCR4 is essentially on all cells, even if at different levels. So we knew that the migration of cells was abrogated by pertussis toxin, which is an inhibitor of a class of G protein receptors. So we tried the cells which are CXCR4 knockout, and cells which are CXCR4 knockout are not attracted by HMGB1, but are still attracted by other chemoattractants like PDGF. So we thought that CXCR4 is the receptor for HMGB1, but then we could not find HMGB1 binding to CXCR4. And so the known ligand for CXCR4 is a chemokine which is called SDF1 or CXCL12. And actually, when we use the HMGB1 as a chemoattractant and then put in an antibody against CXCL12, the chemoattraction was abrogated. So this shows that CXCL12 is involved in the chemoattraction produced by HMGB1. And we showed that CXCL12 and HMGB1 synergize with each other. So a certain concentration of HMGB1, which is not chemoattractant, together with a concentration of CXCL12, which is not chemoattractant, together produce a chemoattractant effect. And so this is due essentially to the fact that the proteins form a labile complex, which can be seen fairly easily by NMR. So if you put together the two proteins, you do see shifts in the frequencies of resonance of several amide protons. And if you map the regions of interaction, CXCL12 is almost completely embraced by HMGB1, and it binds to both box A and box B, mostly to box A, but also to box B. So at this point, we thought that the chemoattractant was the complex between HMG1 and CXCL12. So we went to a model of acute damage and immune cell fraction, which is cardiotoxin injection into a muscle. So if you inject cardiotoxin, this lyses some muscle cells, and if you have cell lysis, HMG1 is released. And so what happens when you destroy a little bit of muscle is that a lot of inflammatory cells come in. So if you stain these two situations for HMGB1, you see that HMG1 is in the nuclei of muscle cells, but after damage, it's in the nuclei of muscle cells plus the nuclei of inflammatory cells, but also, I don't know if you see here, but you see some HMG1 in the cytoplasm of the muscle cells, and the muscle cells actually start also releasing HMGB1 actively. So what happens here? So can we stop the influx of inflammatory cells? So first of all, we can detect the complex by using hybrid ELISA, where we capture CXCL12, and then we develop with an antibody against HMGB1, and we see that uninjected animals have very little complex in the muscle. Then the complex starts appearing, and you don't see any signal with HMG1 alone or CXCL12 alone. We also, in this model, can inject anti-HMGB1 antibodies, and then we block the arrival of monocytes and granulocytes, neutrophils, almost completely. And we get the same result with glycyrrhizin that we had identified as a natural component, a small molecule, which is an inhibitor of HMGB1. So the situation at that point was complicated by the fact that our results were completely non-comparable with results of Kevin Tracy, who had shown that the receptor for HMGB1 was TLR4. So his results looked okay. Our results, we were sure, were okay. So there was something that was different between our protocols. So it turns out that HMG1 has 3 cysteines, which is already by itself unusual because histanes usually come in pairs. So box A has 2 cysteines that actually are just in front of each other and can form a disulfide bridge. The third cysteine is non-paired and is always a thiol. Does not form a bridge with anything else. So in Kevin Tracy's experiments, the third cysteine was really important because if you blocked it, there was no effect of HMGB1. While if you didn't block this cysteine, HMG1 caused the TNF production, and the identified receptor was TLR4 because macrophages that were TLR4 knockout did not produce TNF. Okay. So at this point, we thought that the difference was in the redox status of the cysteines. So it turns out that our HMGB1, Kevin Tracy's HMGB1 produces or helps produce TNF-alpha and MIP2 by macrophages, while the HMG1 we produced with reducing the histines does not. On the other hand, our HMGB1 was a chemoattractant, but did not induce production of TNF. And the only difference was the use of DTT during the production. So we next showed that our HMGB1 in non-reducing polycleramide gel electrophoresis was migrating a little slower, which is indicative of non-formation of disulfide bonds, while Kevin Trace's HMG1 was migrating a little faster. And if we looked at mass spectrometry, there was just a 2 Dalton difference between the total weight of the protein, which corresponds exactly to the 2 hydrogen atoms which are lost during the formation of hydrogen bond. So to make a long story short, the protein with 3 thiols essentially does not induce the production of cytokines, but has chemotractant activity. The one with a disulfide bond has cytokine-inducing activity, but does not have chemotractant activity. And the one with where the histines are further oxidized to sulfones has neither activity. Then the model we proposed, and I stress that this is still a model, and someone should really try to demonstrate it in the details, but we haven't, is that essentially when you have cytotoxin injection and necrosis of the muscle cells, HMG1 is released as it is in the cell. So it's reduced in the cell because the cell has a very reducing redox potential. The protein acts as a chemotractant, so chemotracts leukocytes from the blood vessel into the damaged tissue. Now, some of the HMGB1 by this time is already oxidized and activates now the leukocytes by binding to the toll-like receptor 4. So now the macrophages that have been chemotracted are also activated and start producing a lot of different things, including ROS, that further oxidize HMGB1 and transform it to the non-active species. So essentially, this is a self-contained story because you attract the cells, you activate them, and then the chemotractin is destroyed. And then you let the system go, and eventually, in some way, it helps clean up the system, and then the tissue can be regenerated. But then, of course, you have to regenerate the system and with the tissue, and this is what we pursued, essentially. So we hypothesized that after having done all of this, so having attracted the cells, having activated them, then the protein would also lead the reconstruction of the system. It was, I would say, efficient to do this. So we first looked at muscle regeneration in heterozygote mice for HMGB1. So the knockout mice die very early after birth, so could not be used. But the heterozygotes have exactly half the amount of HMGB1. And if you use half the amount of a chemotractant, you have half the amount of effect, but also, in general, for a secreted protein, half the amount gives half the effect. So we looked at the regeneration of muscles in these conditions, and we thought that regeneration was delayed. We had less myoglobin mRNA, which is a marker of muscle, after a few days. But early on, we had a reduced recruitment of leukocytes, and we had a decreased activation of satellite cells that are the cells that replace the muscle cells. And we also had, later on, defective angiogenesis. So the whole reconstruction of the muscle was delayed. So at this point, we used a protein which is a mimic of a reduced protein, which we called 3S. So essentially, we took HMG1 and replaced the cystines with serines. So the protein cannot be oxidized. So if we use this protein, we get essentially the same result as injecting the reduced protein, but at least we are sure that the protein can never be oxidized. And in this case, both injecting the reduced protein and injecting the 3S form of the protein, not only the muscle regenerates, but it regenerates faster than in the knockout, than in the wild type. So we accelerate the normal reconstruction of the tissue. And this happens because the satellite cells are more activated, because the macrophages which are attracted are then shifted earlier to a tissue repairing phenotype, so NEM2 phenotype. And HMG1 can also shift cells which have already been polarized to M1 back to M2. And also can activate the endothelial cells to regenerate the blood vessels. So essentially, HMG1 in our model orchestrates the action of different cells in a correct timing so that they all now contribute to regenerate the muscle. And in our idea, when you have acute muscle injury, you release HMGB1. From the blood vessel, you recruit monocytes. The monocytes turn into macrophages, which are now of the M2 type. They help in different ways shape the microenvironment, and HMG1 and macrophages together help reconstruct the tissue. So the same thing was proven in another tissue damage model, which is bone fracture, by our collaborator, Nancha Hao. So essentially, in a bone fracture model where you have essentially the two stumps of the bone kept separated, takes time to repair the bone. But if you inject either reduced HMG1 or 3S HMGB1, it takes much less to repair the bone. But actually, the repaired bone is also of better quality, in the sense that it has a better mineral density and is less different from the initial, from the repaired bone than in the normal situation. So it's one of the rare occasions where we can do better than physiological repair. So the summary for this is that, of course, you cannot have regeneration of damaged tissue without inflammation, and this is written in every pathology text. But HMG1 is important for inflammation, but also for repair, so for tissue regeneration after damage. In muscle, HMG1 recruits macrophages, acts on satellite cells, and polarizes macrophages to a tissue healing phenotype. And injecting some HMG1 is good, but injecting 3S HMG1 is better because it cannot be oxidized. And one shot is enough. And having time, the healing time is more or less halved. And this acts on multiple tissue because it works on muscle and bone, but also works on liver. We did the same thing in liver, which heals in essentially different way, which is not dependent on stem cells. So we came up with the idea that the oxidized HMGB1 causes inflammation, and so long as you have inflammation, you cannot regenerate the tissue. But as soon as you stop inflammation, the HMGB1, which is continually released, is reduced, and now the reduced HMGB1 orchestrates the tissue regeneration, which is very convenient because you have a single sensor of a situation of a tissue. If the tissue is inflamed, it is more oxidative, and the HMG1 is oxidized and keeps on the inflammation. If the tissue is not inflamed, then the normal condition is reducing, and HMG1 is reduced and help tissue regeneration. So, of course, this creates a conundrum valve, because if HMG1 helps the reconstruction of a damaged tissue, probably it's detrimental in cancer, because cancer usually behaves as a damaged tissue and recruits the help of the immune system, and especially of macrophages, as tumor-associated macrophages, to grow. So essentially, we looked at mesothelioma, which is a very aggressive cancer, which is caused by asbestos fibers. And we showed with the help of Michele Carbone, or Michele Carbone showed with the help of us, that the reason why asbestos causes mesothelioma is that asbestos kills the mesothelial cells. Of course, this is not the entire story, because if it kills the cells, it cannot cause cancer, because cancer is overgrowth of cells, not cell killing. But the killed cells release HMGB1, the HMGB1 recruits the macrophages, and the macrophages help survive the remaining mesothelial cells, and eventually a clone appears that is cancer. So this takes a long time, often decades, but HMG1 is essential for this, and we could show this in a mouse model. So not only the HMG1 is released by dead mesothelial cells, but it turns out that the macrophages that are recruited also secrete HMGB1, in addition to the cells that become cancer. So in mesothelioma, both the cancer itself and the macrophages secrete HMGB1. So at this point, since HMGB1 helps the tissue grow and regenerate, it should help HMGB1, it should help cancer proliferate. And in fact, in this case, secreted HMG1 should be a cancer for a target for cancer therapy. So we created the model, mouse model of mesothelioma, and we showed that mesothelioma produced in this cancer model contains in the cancer up to 30% of macrophages, which is exactly similar to what happens in the cancer of mesothelioma patients. And all of this, not all, but 90% of these macrophages were of the tissue healing phenotype, or M2 phenotype. Okay, so at this point, we tried with an anti-HMGB1 antibody, and this has a small effect on the development of mesothelioma. But then we tried BoxA. And BoxA is the fragment of HMG1 that you see here in square, and it binds to the receptor of HMG1, but does not activate them. So it's a false ligand, essentially. When we used BoxA, we got a result which is essentially fantastic. So a lot of mice survived much longer, but actually a few mice even survived for a long time. So overcame the tumor. But this was done in immunodeficient mouse. So we said, what happens in an immunocompetent mouse? Does it work even in an immunocompetent mouse? And in fact, turns out that mice treated with BoxA have a strong reduction of a tumor, and a lot of them survive and actually live forever, for a long time. They have no remaining tumor. So essentially, this was much better than the immunodeficient mice. So since the difference was between immunocompetent and immunodeficient mice, we thought that the immune system might play a role in this. And in fact, what we did was to re-inject the mice that had no surviving tumor, and we measure the tumor by the amount of photons that are produced by tumor cells that have luciferase in them. We re-injected the tumor cells, and the tumor cells were rejected in less than a week. So essentially, the mice were immunized against the tumor. Then we took the T cells from the spleen, and we pulsed them with a luciferase peptide. So there were T cells that were recognized as luciferase, but in our case was, of course, a new antigen in the tumor. Then the T cells should proliferate. And this is what we observed. So essentially, in the naive mouse, there was no cell that was proliferating. In the control mice, the mice that had been injected with the tumor only, there were no cells that were responding to the look peptide. In the mice that were treated but did not respond, there was no response. But in the mice that had responded, there were a lot of cells that responded to pulsing with the peptide. And so the T cells were recognizing the luciferase peptide. So the whole thing we showed, so the rejection of a tumor depended on T cells. And in fact, if we deleted the CD8 cells from the mouse, there was no response to BoxA. So the mouse were not cured by BoxA. So at this point, we asked what was the likely reason, and we spent a lot of time on a false thread. It was already known that tumor cells challenged with chemotherapeutic agents go into apoptosis. And upon going into apoptosis, before they apoptose, they release three key molecules, which are ATP, HMGB1, and calreticulin. So calreticulin is an eat me molecule. So when it's seen by the immune system or macrophages, promotes the phagocytosis. HMGB1 does all the things I told you. And ATP is essentially a find me molecule, so helps the macrophages find the tumor. In this case, when the tumor cell die, and having released these three molecules, they essentially are recognized by immature dendritic cells that eat the dead cancer cells, and then expose the antigens to T cells, first the CD4 cells, then the CD8, and the clones emerge of T cells that recognize the tumor and then kill the tumor by immunological responses. So the tumor is essentially killed twice, first by the chemotherapeutic agent, and then by the immune response. And Guido Krömer, who discovered this type of death, which is called immunogenic cell death, argues that any chemotherapeutic that is effective works twice. First, to kill the tumor cells, but then also to elicit an immune response. So we thought that this was the cause, but in fact, it wasn't, because essentially the immunogenic cell death activates what is called the unfolded protein response, which is a response of endoplasmic reticulum that gets essentially clogged with unfolded proteins. The unfolded proteins activate responses through three receptors in the membrane of ER, and this leads to release of HMGB1, calreticulin, and ATP. It turns out that our cells exposed to BoxA, the tumor cells, actually release calreticulin. And you can see calreticulin on the surface of the tumor cells. They release HMGB1 and they release ATP, but the cells don't die, never. And also the response is a little bit different because the ICD activates all three arms of unfolded protein response, while our treatment, BoxA, activates only one. And mostly, since the cells don't die, there is no way the dendritic cells could essentially see the apoptotic bodies, which are necessary for the dendritic cells to expose the antigens to the surface. So we thought that something else was happening. And we thought about a system which was already known because it has been investigated by Weissman for a long time, which is the exposure of CD47. CD47 is a don't eat me molecule, which works by interacting with SERP-alpha, which is a molecule on the surface of macrophages. So macrophages that see cancer cells with CD47 don't eat them. And there is no antigenic response against these cancer cells. But if you now block either CD47 or SERP-alpha with antibodies, then there is no recognition of the don't eat me signal. The macrophages eat the cancer cells, now expose the antigen on the surface, and you have a T cell response. Okay? So we looked at what was happening in a macrophage phagocytosis assay. So here, the cancer cells are green, and the macrophages are the small gray, dark gray things that you see going around the tumor cells. The macrophages are smaller than the tumor cells. But look what happens. Look at that small cancer cell. A macrophage eventually arrives and eats up the cell alive. It's like you eating a sandwich which is about 120 kilos in one gulp. And the number of the cells that were eaten up by macrophages increased with the dose of box-A. So essentially, box-A was inducing not apoptosis, but phagocytosis of cancer cells, which then were adequate fodder for the macrophages to expose antigens. It's an alternative pathway, but again, the antigens are exposed. And we could show that CD47 actually goes down, not by much, on the cancer cells by exposure to box-A, but a 30% decrease had already been shown by Weissman to be sufficient for phagocytosis by macrophages. We could see that the reason why the cancer cells were phagocytosed was the disappearance of CD47 from the surface. And here what you see is the PLA, which is a way to see the contact of two molecules. Here is the PLA between CD47 and CXCR4, which is the receptor for HMGB1 and for box-A. And you can see that initially there is a lot of contact between CXCR4 and CD47 on the surface of cells, but if you treat them, this contact goes away. While if you permeabilize the cells, initially you don't see the molecules inside the cells, but now all the molecules are inside the cell. So essentially what happens is that CXCR4 is bound by box-A, goes inside the cell, bringing together with it the molecules of CD47 to which it's already in contact, and the cell now ends up phagocytosed by macrophages. Of course, CD47 internalization is CXCR4 dependent, because if we inhibit CXCR4 with an inhibitor of CXCR4, CD47 is not internalized. But we also showed that the whole thing works not with box-A, which is an exogenous molecule. Essentially, we hit upon this mechanism because we were using box-A for different reasons. But it also works with the natural ligand of CXCR4, which is CXCL12. So if you give CXCL12, CD47 decreases, the CXCR4 is internalized and brings with it CD47, and cells are phagocytosed by macrophages. So this works in a different model of tumor, so it's not only malignant mesothelioma, it's also cancer, colorectal carcinoma. And so the conclusion here is that we have that CXCL12 promotes tissue repair and tumor growth, but also CXCL12 in some way interacts with CXCR4 and also promotes the phagocytosis of some tumor cells. The effect is not due to phagocytosis per se. The macrophages don't eat up all the tumor. And in fact, we cannot find easily macrophages in the tumor that have eaten up a tumor cell. But even just eating a few tumor cells, they would expose the antigens to the surface and in some way now activate adaptive immune responses so that eventually you see the emergence of T cell clones that kill the tumor. So since the CXCR12 is produced by the tumor cells themselves, it turns out that the tumor cells recruit the macrophages to help their growth, but a few macrophages also eat the tumor cells. So you have two opposing effects. On one hand, CXCL12 promotes tumor growth, but on the other hand, exposes the tumor to the surveillance of the immune system, and eventually some of the tumors are rejected. So this probably happens all the time in every organism or every mammalian individual. And so some of the tumors are probably rejected because the macrophages see the tumor cells. Except that the tumors that end up killing the patient are the ones that could evade the system. So in principle, this is a system of immunogenic surrender of cells to the immune system. The cells themselves identify to the immune system. The macrophages eat up the cells, and then you have an immune response. So the ideal thing here is to activate the immune response without actually helping the tumor. So we are trying to see what happens if we treat the tumor with CXCL12. We expect that some mice will do worse because CXCL12 will help the tumor grow, but some mice will do better. They will reject the tumor. We have done this experiment twice, and we had a clear result as we expected the first time, a very fuzzy result. The second time, we are waiting to redo it the third time. But we also think that this is a check and balance mechanism that is active always. So if we could activate the immune response branch better than the tumor growth branch, we could do probably service to any individual who is having a small tumor. So what we are doing is now proving the hypothesis that immune surrender makes antigens more visible for checkpoint therapy, essentially because if you show the antigens, then immune checkpoint therapy can activate the T cells to kill the cells that expose those antigens, but you have to recognize the antigens first. So the two things should be complementary. We want to screen for small molecules that bind to CXCR4 and promote CD47 internalization, but do not promote tumor growth like CXCL12. We think it's possible because CXCR4 is a G protein, and G proteins can be tweaked by small molecules to do different things with different molecules. And then we are, as I told you, evaluate CXCL12 itself as immune adjuvant, but will never be used as an anti-tumor therapy, but would simply prove the point. And this is the last slide. I'm sorry, it took a little longer than I thought. This work, the last part of the work, the immunogenic surrender work, was done mostly by Rosanna Mezzapelle, who is actually sitting there. And we had the help from HMG Biotech, which is the company that I founded and produces all the molecules that the proteins that I told you. This is also a disclaimer, even if so far it did not produce any significant financial gain. We had help from Anna Rubatelli, who actually helped us show that this was not immunogenic cell death, and Vittorio Colantuoni, who initially proposed it was immunogenic cell death, but then we could disprove the idea. And Anna Mondino, who is the immunologist, helped us frame some of the experiments that were critical. This was funded by AIRC and Fondazione Buzzi and some internal grants at Università degli Studi del Sannio. And thank you for your patience.
Okay. Thanks, Marco, for the beautiful presentation.